this video, we will learn about the role of raw material used in the process of photosynthesis through experiments. Objective: to demonstrate that glucose formed during photosynthesis is converted into starch in plant and to observe the effect of light on this process. So, objective of this experiment is to demonstrate that glucose formed during photosynthesis is converted into starch in plants and to observe the effects of light on this process. Materials required: 2 D-starch potted plants, plant A and plant B, dark enclosure, sunlight, boiling water, alcohol, warm water, iodine solution, glass plate, and labels for the leaves. Procedure: take 2 D-starch potted plants and label them A and B. Then second step is keep plant A in dark place while plant B in sunlight for 4 to 5 hours. Then after 4 to 5 hours, pluck one leaf from each plant A and plant B, then mark them as A and B. Boil both leaves separately, first in water, then in alcohol. Wash them with warm water to remove alcohol. Place the leaves on a glass plate and pour few drops of iodine solutions on them. Leaf B turns blue-black, but leaf A does not. This is because plant B starch is formed by photosynthesis as it was kept in sunlight. Plant A was kept in dark, so no photosynthesis occurred and no starch was formed. So the procedure is, in the first step, we have to take a 2 D-starch potted plant and label them as A and B. Plant A in dark place while plant B in sunlight for about 4 to 5 hours. Then after 4 to 5 hours, pluck one leaf from each plant A and B, then mark them as A and B. Then we have to boil both the leaves A and B separately, first in water, then in alcohol. Then wash these leaves with warm water to remove alcohol. Then we have to place these leaves on a glass plate and pour a few drops of iodine solution on them. Then leaf B turns blue-black, but leaf A does not. This is because plant B starch is formed by photosynthesis as it was kept in sunlight, while in plant A, which was kept in dark, showed no photosynthesis, so no starch formation was occurred. So the conclusion is, the experiment illustrates that starch is formed in plant B due to photosynthesis in the presence of sunlight, while plant A kept in dark does not produce starch. So this is the whole experiment. We will take 2 D-starch potted plant, plant A and plant B. Plant A is kept in dark while plant B is kept in sunlight. Then we will pluck leaf from each plant. Then we will boil them separately, first in water, then in alcohol. Then wash them in warm water to remove alcohol. So this is leaf A and leaf B. We will put this leaf on a glass plate and pour few drops of iodine solution. So as we can see in plant A, no change in color because starch is not formed, while in plant B, change in color after pouring few drops of iodine solution because starch is formed. So the experiment illustrates that starch is formed in plant B because it changes its color in blue and black due to presence of starch, while plant A, which was kept in dark, does not produce starch. Nutrients formed from glucose: sucrose and starch, proteins and fats. Sucrose is used for plant growth and also stored in fruits. Starch is stored as a food in different parts of the plant. So sucrose stored in fruits, sucrose stored as a starch in roots, and sucrose is transported in phloem tubes to all parts of the plant. Next is protein, made from simple carbohydrates and nitrogen. Fat formed from simple carbohydrates. Now we will talk about heterotrophic plants. Based on method of obtaining food, Heterotrophic plants may be parasitic plants, saprophytes, symbiotic plants, and insectivorous plants. First of all, we will talk about parasitic plants. So, the organisms that live on other organisms for food are called parasites. So, the parasitic plants are organisms that live on other organisms for food. Parasitic plants obtain their food from some other green plants. The plant that provides food is called host plant. So, the parasitic plant obtain food from other green plants. And these green plants photosynthesize their food. And these parasitic plants obtain their food from these green plants. And the plant which provide food is called as host plant. Now, parasitic plants are divided into two types, total parasitic plants and partial parasitic plants. Total parasitic plants are like dodder, also called cuscuta, completely depend on host plant for their food. While partial parasitic plants can synthesize their food but depend on host plant for water and mineral. For example, mistletoe is a partial parasite on mango and mahua plant. First one is total parasitic plants like cuscuta, which completely depends on host plant for their food, while partial parasitic plants can synthesize their food but depend on host plant for water and minerals. For example, mistletoe, which is partial parasitic plant on mango and mahua plant. So, this is dodder, which is cuscuta, and we can see here this is cuscuta, and cuscuta is completely dependent on this green host plant for their food requirement. And this is mistletoe. Mistletoe is a partial parasitic plant. Next one is saprophytes. Saprophytes are non-green plants which obtain their food from dead and decaying plants and animals. Fungi such as molds and mushroom and some bacteria are saprophytes. So, saprophytes are non-green plants, and these plants obtain their food from dead and decaying plants and animals. And examples are fungi such as molds and mushroom. So, molds and mushroom are non-green plants which obtain food from dead and decaying plants and animals, and these are saprophytes. I hope this topic is clear to you. Thank you.